So you touched a little bit on the big three or the big four, and that's typically what a limited test might look like. But to your point, and some of the things you said earlier, so many exciting things are happening in the lung cancer space right now. So I want to
Mm.
talk about some of those lesser known and HER2, MET, RET, NTRK, KRAS G12C. You brought that up a little bit ago, and then even the PIK3CA. This is something that's kind of emerging in the squamous space. So I don't know where you want to start or which one you want to start with,
Yeah.
but talk about some of the exciting things that have been happening in that space.
Yeah, I wish we had 3 hours because
I
the
know.
list gets longer every day in terms of, I mean, we've had something on the order of 5 or 6 drug approvals in lung cancer in the past 6 months. So it's just insane. Yeah, as you mentioned, EGFR mutations and ALK rearrangements and ROS rearrangements were sort of the leading edge. They were like I said, chapter 1, 2, and 3 of molecular profiling in lung cancer. And if you would have asked me how many chapters are going to be in this book 5 years ago, I would have said maybe 4 or maybe 5. And as you've mentioned, that we are now learning more and more about other mutations outside of just EGFR, ALK, and ROS that can be identified in lung cancer and can help select tailored therapies. And the nice thing about this is that the mutations are mutually exclusive. It makes it easy. If you have one, you don't have the other. So you never have two mutations. It's a nice story. It's a clean story. And oftentimes this is represented pictorially by a pie graph, and every slice of the pie is a different mutation. So as mentioned, EGFR, ALK, and ROS, chapter 1, 2, and 3, but we've got about 6 other chapters. Some of the more are less common, but still there. I mean, there are a lot. These are less common than EGFR, but certainly I see a ton of these patients. So, looking at things like RET rearrangements. RET rearrangements are the new darling child for lung cancer. The RET rearrangements are around 1 to 2% of all lung cancer. If you can identify a RET rearranged lung cancer, we have a new drug approved called Selpercatinib. It's an oral therapy. It's well tolerated. It's exquisitely can attack the tumor. It elicits responses for patients who have brain metastases, so it crosses the blood-brain barrier. And that drug is approved recently. We have another drug called Pralsetinib for RET rearranged lung cancers. It's not yet approved, but also showing again oral therapy showing good response rates to patient oral therapy eliciting good response rates. Um, Outside of RET, then you have other mutations called MET exon 14 skipping mutations. I'm happy to say that again, MET exon 14 skipping mutations. Yes, there's a mutation called that. And that again, if you have a MET exon 14 skipping mutation, this is yet another mutation that has a new approved targeted therapy, a drug called Capmatinib, oral therapy, well tolerated, targets selectively against that mutation. Other mutations like NTRK fusions. NTRK fusions are an interesting story. It's a very rare in lung cancer, but we still find them. The interesting thing about NTRK fusions is that they are seen in other tumor types, and they're common in rare tumors, and they're less common or infrequent in common tumors. And the trial that basically got the drug Larotrectinib approved was an interesting trial design. It was an umbrella trial design that essentially looked at NTRK fusion patients irrespective of where the tumor, what type of tumor you had, and every patient got Larotrectinib. And again, this is another approval that we have. And then 2 more that I want to mention. The first is HER2 mutation. This is a biomarker that we know is relevant in breast cancer. And we have many drugs approved in the breast cancer world that are HER2 positive, at least by a different type of assay. But HER2 mutations are now found to be relevant in lung cancer. And we had data just 5 weeks ago, 6 weeks ago, I guess, sorry, 2 months ago. So 8 weeks ago at ASCO, looking at a new drug called Trastuzumab deruxtecan. It's a new class of drugs for lung cancer. It's an antibody drug conjugate. It's an infusion. but it specifically targets patients that have HER2 mutations. And again, giving this drug, it looks really good. The drugs elicit meaningful responses. They're well tolerated. And then finally, KRAS G12C. This is yet another mutation that 5-10 years ago, I would have said there is no way we're going to be able to drug this mutation for many reasons that I won't get into. But biologically, it's a very hard mutation to target. And we know that for patients with KRAS G12C, there is not yet a drug approved, but there's a drug that's moving fast, an oral therapy that can again elicit meaningful responses and really change the way that we think about KRAS mutations because they were thought to be so undruggable. So for many of these ones that I mentioned to, we have approved therapies. For HER2 mutations, Trastuzumab deruxtecan is not approved yet. For KRAS G12C, the Amgen drug, Amgen 510, is not approved yet. But I think it will be in the next 6 to 12 months. And it's just a very exciting time. And I think all of this, Danielle, I think this truly underscores the importance of comprehensive genomic profiling, next generation sequencing. We cannot just get away with doing EGFR and ALK. And you just got to, I mean, I can't tell you how many second opinions I see where I look at what the genetic testing was done on the patient, and not to throw shade on any of the private practice people because they're great and they're working in the best interest of the patient. But the data is so quickly evolving, it's tough to keep up with. But I can't tell you how many patients I've had where they show me their molecular testing and I say, what else did you have done? And where's your liquid biopsy test? And unfortunately, they just had 3 genes tested, EGFR, ALK, and ROS. And you're missing 7 or 8 other genes that either have approved therapies or therapies that we have trials for. So, and the last thing I'll say, and then you can shut me up, is that the conversation of it's so hard for me to describe the difference in the conversation that goes down. It's not good or bad, but just different. The difference in the conversation that goes down when we're offering targeted therapies versus offering chemotherapy or immunotherapy. They're entirely different conversations. And we have to know this information up front so we know how to tailor that conversation the same way we're tailoring the therapy. I mean, it's a very different conversation that goes down when it's, hey, you're going to be on a pill and I'm going to see you in 2 weeks and you're going to call me and tell me how you're doing, versus, hey, you need to get a port put in and we're going to do an IV on you every 3 weeks. And I'm not saying that there's anything wrong with that, because we do that for a lot of our patients and that's okay. But I think in the end, we have to know this information and we have to do the comprehensive genomic profile. And it can't just be three genes. If you learn anything tonight, just test, don't guess, get the comprehensive testing done and do next generation sequencing, both in tissue and in blood.
I mean, and I know she's like doing this and like little dances in her head crazy, right now. Right? Yeah. I mean, but I think this is why, we're constantly sort of browbeating people about this testing and why it's important. And you couldn't be saying it more perfectly or eloquently. And I think this points back to none of this would have happened without clinical trials. Even if there's not a drug available currently or a target based on what your biomarker testing shows, there might be something in clinical trials that is showing huge promise. And that might be your best first line treatment, right? As opposed to, sitting in the infusion chair. So
yeah, and can I just say one thing about clinical trials before we move on?
Yeah.
And that, people think like, well, if I'm going to a clinical trial, I have a 50% chance of being a guinea pig. And what I tell patients is that couldn't be further from the truth. All of our trials generally don't have a placebo arm. All the trials ensure the delivery of the therapy being tested. And I just want to be very clear that, people think clinical trials are a bad word, or that you're at the end of the line, or that there are no other options. And it couldn't be further from the truth, given all the science that we've really, truly tried to leverage and all the drugs that are available. Most of the trials now are designed specifically for patients that does not involve a placebo or control arm. So you're 100% assured of getting the drug of that we're testing.